Thank you, everyone, for having me. So I'm going to tell you a little bit about some work we've been doing in my lab where we use Compound Discoverer in concert with a number of other tools for identifying compounds, in this case in house dust. And so I'm an environmental chemist primarily, so most of my work is trying to answer these questions. How can we address both human as well as ecological chemical exposure? And so one of the big problems that we face is in environmental chemistry is that we take a really complex mixture. Actually, I can use maybe just the, yeah. So we take a really complex mixture of chemicals that we might apply in industry and commerce, and we mill this through ourselves, our agriculture, our homes, our wastewater treatment plants, and we take this and transform it into a much more complex mixture, which then we might have effects. And so we're trying to map out these chemicals that are most relevant for exposure out of a very, very complex mixture, and so within chemical space. And so the challenges we face include incomplete information about what is actually being used, especially in the US. We don't really get a lot of information on chemical use and production. Also biotransformation and other things making prioritization difficult. And so you can kind of see this is just a map of all of PubChem in the gray dots on basically principal component space for just illustrating chemical space as a function of molecular descriptors, and then how different databases like the EPA's DSS Tox, Drug Bank, HMDB kind of map onto this. And you can see some trends that not all of these databases overlap the same chemical space. So we have some challenges related to this as well. So as we implement non-targeted analysis in my laboratory, we're doing this using a Orbitrap fusion Lumos system, the Tribrids. And just a note about why we use this instead of the QE platform. The QE platform's great, but critically, it's actually the mass accuracy that we get with the Lumos, especially with the easy IC internal calibration. We can routinely get less than a part per million mass accuracy without really having to do much. And the critical point about this is that it's stable over many days. So we can calibrate and then set the easy IC, and we can rely on less than a ppm mass accuracy. Also, of course, the MS3 capability and the ion funnel, which of course you'd also have on the QE HFX, gives really good sensitivity. We're also running relatively long runs, so we do about 45-minute LC, ESI, as well as APCI runs on this. We have started to use the AcquireX workflow. So this has actually really changed the way that we do a lot of our experiments. So we did some testing with some selected house dust samples. I won't show this data other than just to mention that when we do our standard DDA, if we do 3 replicate injections with just intensity-dependent precursor selection, we're getting about 66% MS2 coverage, okay? And this is just injecting the same sample 3 times and letting the DDA do its thing. If we implement the AcquireX, we're increasing this up to about 84%, 85% MS2 coverage. And so there are still some challenges. There are compounds that just aren't being selected by AcquireX, and we're not sure why that is. We've been talking to some folks at Thermo about that. But this has actually really increased our coverage and allowed us to use DDA instead of as a primary strategy, and still believe that we're getting a pretty wide swath of compounds identified. So I wanted to show this slide just to kind of give a picture of why mass accuracy is important. I mean, I think we all appreciate this, but it's kind of nice to see it with data. And what I did here is I took PubChem, all of PubChem. It's about 65 million organic molecules. So how many molecular formulas is this? This turns out to be about 2 million unique molecular formulas within PubChem across the entire database, up to 1,200 Daltons. And what I did is I binned all of these compounds into 1 Dalton windows. And I said, okay, at any integer mass, what mass accuracy is required to uniquely identify a particular molecular formula? And so what this is a plot of the percent unique formulas at the specified mass accuracy as a function of molecular weight. And what you can see is that at 10 ppm, very, very quickly you fall off here, and so you get a very, very low percentage, and it approaches 0 as you enter sort of the main space that we're interested in with respect to compound identification. So almost no molecular formulas are unique at 10 ppm. But what's interesting is even at 1 ppm, you can see that we do pretty well up to about 250 Daltons. And then after that, we get into this sort of valley here where even at 1 part per million, only about 10% of the formulas are actually unique at that mass accuracy at 1 ppm down here in the 4 to about 800 molecular weight range. Okay, so the answer is how much mass accuracy do we really need? As much as we can get. Okay, and so we can't just rely on mass measurement alone to give us these formula assignments. This is the same plot, but now this is actually a histogram or a distribution of the compounds, right? So this is all of the 65 million molecular structures, right, mapped here. And so you can see that we overlap pretty well, at least in low mass for at 1 ppm. So 1 ppm is pretty good, but it's not quite good enough. Okay. So we're doing compound identification, and so in environmental chemistry, there was this paper that came out in 2014, which laid out a confidence level scale, level 1 through 5, for how well we identify our compounds. Level 1 would be a confirmed structure by using a reference standard, so we can compare against our standards. This is everybody knows how to do this. Probable structure level 2. This is primarily library spectral matching. Okay, with MS2 libraries, all right? Level 3 is where we don't have library matches, okay? And so this is actually where the hard work begins. Level 4 is when we can only get an unequivocal molecular formula. I just showed you how that can be difficult even with 1 ppm mass accuracy from mass measurement alone. And then level 5 is only when we have a exact mass of interest. So let's take a little bit of a deep dive into how we do a good job with level 2 identifications and how we address that. What we've done is actually we got a little impatient before MZ Vault 2.1 was announced. We wanted to try to maximize the number of MS2 library spectra that we could access using Compound Discoverer. And so what we did is actually, so MZ Cloud is pretty easy, right? We access that directly through Compound Discoverer, but we also have MassBank and the NIST MSMS databases that we'd like to use. And so what we did is we figured out the database format for MZ Vault, and we actually wrote a script, custom R script, that basically takes these data and translates them, first of all, processes them through Sirius, which I'll show you that algorithm in a minute, to annotate all of the MSMS spectra, high-resolution MSMS spectra in both MassBank as well as NIST, annotate all the fragments for formula, and then load those into a MZ Vault database that can then be put into Compound Discoverer, and we can search our spectra in an integrated way. So if we look at what we end up with about 22,000 compounds. So, and this MZ Cloud number, this is basically an intersection. This is the number of compounds in unique compounds in each one of these databases, and these are the overlaps. And what you can see is that, at least this was in 2017, NIST had the largest number of unique compounds represented, followed by MassBank and then MZ Cloud. MZ Cloud, of course, is getting bigger, as we heard yesterday. But what's interesting about this is that you can see that most of the compounds in each one of these databases are actually unique to those databases. So there's not as much overlap among these spectral libraries as you might think. So putting them all together gives us a lot more compounds that we can select from. The problem is that the data is of varying quality. And so not all, well, first let me just tell you how Sirius works. This is how we're annotating all of the library spectra that we get from MassBank and NIST. These are actually not very well curated in some cases. What Sirius 4.0 does, this is Sebastian Bacher's group has developed this. Basically what we do is we take our MS isotope information as well as our high resolution MS2 information. We rank the formulas that are possible. Now, in our case, for these library spectra, we know the formula a priori, so we give that formula to Sirius, and what it does is it then creates a fragmentation tree which annotates all of the fragments, assuming that all the fragments must be a subgraph and a subformula of the precursor. And so what this allows us to do is essentially assign known mass errors for each one of the fragments for every spectrum in every library. And so why that's important is that if you take a look at the mass accuracy distributions across these databases, they're not exactly equal. And so this is actually a plot of mass accuracy versus density for all of the Orbitrap and FT data. There's 345,000 or 344,000 Orbitrap and FT spectra in NIST. There's about 10,000 QTOF spectra in NIST. And if you look at the mass accuracy distributions for the Orbitrap and FT data, it's all at less than 10 ppm for all the fragments. But of course, the QTOF data, you run all the way out to 45 or so part per million. Now, so the question would be, if you use these spectra with Orbitrap data as your query, what do you set your mass error to? Well, so in this case, because we were able to annotate every fragment in every spectrum with a known mass error, it turns out that the MZVault library format has a mass error column that you can enter for every peak. And if you use the HiChem high resolution matching algorithm, it actually looks in that column and uses that mass error, that known mass error, to, in real time, update your mass accuracy. So you don't have to worry about what to set for every spectrum for your mass accuracy. It does this automatically. So that's the way that we use the NIST and MassBank library data. One thing to note is that especially in NIST, there's a lot of unit resolution spectra, things like ion trap and triple quad. We just set the mass error for each one of the fragments in those spectra to 1 Dalton. And so then that allows us to use that data as well in our searching. So this is actually MassBank, and turns out the mass errors for the Orbitrap are a little better because a lot of these are recalibrated. The QTOF data is still pretty variable. Okay, so we've applied this to house dust analysis. Why do we care about house dust? We're interested in the indoor environment as a source of exposure. There's lots of compounds, consumer chemicals, personal care products, etc. House dust accumulates things like phthalates, plasticizers. As I'll show you, surfactants are actually a really important class of contaminants in the indoor environment. Flame retardants, and then compounds that are in food packaging and other kind of plastics. So we have some overlap with the extractables and leachables community for at least the indoor environment exposure here. So the way that we've implemented this for our library searching, we take our raw files from our Lumos, we process them through Compound Discoverer 3.0. This does all the retention time alignment, peak detection, componentization, etc. This allows us to get our MZ Cloud and NIST and MassBank library match scores all together. Then we've written a few other scripts. First of all, we use RAM clusters. So this is actually a really nice algorithm that was written by Corey Brockling at Colorado State. And so this allows us to consolidate any kind of replication that's still present in the Compound Discoverer feature list. And I'll show you that. So that lets us get rid of things like in-source fragments, adducts, and isotopes that still remain. We wrote another script called CD Consolidator, and this takes the RAM cluster results, merges all of the library results together, picks the best library search, and then also does a rigorous search for in-source fragments. So basically what we do is for every compound that has MS2 data, we take a look at that MS2 spectrum, and then we look for any of the MS2 fragments in a window around the precursor within the MS1 data, and then pull those out and basically call them in-source fragments. So this allows us to consolidate. In this case, we had about 34,000 Compound Discoverer compounds for our data set. We consolidate this down to about 12,000 dereplicated compounds after these tools. And then we have a CD data extractor that's also written in R that pulls out all the MS and MSMS spectra and then puts those into our in silico fragmenters, which I'll show you in a few minutes. Okay, so here's our Compound Discoverer workflow. It looks pretty similar to some of the ones that we saw yesterday. Component detection, grouping, and then we farm out the spectra to things like MZVault. We have some mass lists, custom mass lists that we use based on compounds that we know might be present. We're doing some isotope pattern scoring, looking for things like brominated and chlorinated compounds, and then we annotate. In our experimental design, in this case, we had 190 house dust samples that were collected from a cohort of mothers and children in Durham, North Carolina. The great thing was we had metadata here for things like socioeconomic status, education level, the type of housing, how often they vacuum their house, diet, lifestyle, thyroid function. There's a whole bunch of metadata that we're able to collect for this cohort. So it's a very rich cohort. We did ESI POSNEG and APCI / APPI POSNEG. We ran every one of these samples 4 times, basically. And then we had a bunch of NIST standard reference materials and blanks, et cetera. So it's a huge data set that we ran through here. We talked a little bit about metrics of how we process this and how fast this can or cannot be. In this case, we had a 20-core, 200 gigabyte RAM virtual machine that we ran this on. It took 177 hours. So this was a set for each one of these data sets. Actually, it took quite a bit longer for the APCI. So 7 days, 9 hours, 47 minutes, 261,000 features collapsed to 34,000 compounds. When we use our RAM cluster and our CD consolidator algorithms, we get this down to about 12,600. And then we start to look at the library hits. Our total library hits here was 872. But what's interesting is that we find that if we also have a validator step in our CD consolidator script that goes through and it makes sure that the exact mass matches your candidate formula with known the adducts that were specified, or it goes through and it looks for things like water losses or ammonia losses and tries to rationalize that. But 634 of that 872 were what we call valid. So they actually matched what we expect for the precursor. So if you look at the MZ Cloud only hits, so if we only had MZ Cloud, so we got 174 unique to MZ Cloud, we had 176 unique to MZ Vault. So you can see that this MZ Vault included our NIST and our mass bank spectra. And you can see that they're quite complementary. So the shared library hits was about 284. So if we used only MZ Cloud, we would have gotten about 450 hits. If we used only MZ Vault, that is NIST and mass bank, we would have gotten also about 450 hits, but they would have been different. So it's important to use both of these. So that really helped us with our hit rate. One thing I'll note, this is how we do the dereplication. So here's just an example. This is actually a octylphenol polyethoxylate surfactant oligomer. And if you take a look at the spectrum from compound discovery, you can see lots of adducts here. You get a lot of ammonium, sodium, and potassium adducts for these compounds. You also see the M+H. But then there's a lot of other things in here. And a lot of these peaks actually got called as unique features, but they turn out to be both in silico fragments as well as adducts. So you can actually see in several cases here, we can map out what these are actually unique. These are compounds that were called compounds by Compound Discoverer, but they are actually replications of the different adducts. And so when we do CD or when we do RAM cluster, this actually collapses into a single feature. So it allows us to pull out a lot of the stuff that's essentially noise that's still present in the compound list. And you can look at the peak areas here, and you can see a lot of these are actually really small, small peak areas relative to what we call the parent there. Okay. Another thing I'll note, problem children in Compound Discover, quaternary ammonium salts. Okay, why are these problem children? Because they don't give adducts. They're charged intrinsically, right? So they're quaternary ammonium, so they're cations. The problem is that if you have to give a base adduct that you to Compound Discover so that it assumes some kind of adduct, and then it'll look for other adducts as well to try to figure out which one's correct. In this case, there are no adducts. You get a single peak. And so when Compound Discover sees that single peak, it's going to assign it as M+H. It's actually not. It's an M+. And so as you can see, in the MS1 spectrum here, it's actually giving you this red line where the peak should be, but it's actually 1 Dalton lower. All right. So if you look at it, now of course, why we were able to identify this, because it was actually in our NIST MSMS database, and so the spectral library gave a very nice match here to this benzyl dimethylsterol ammonium cation. But if you look in your annotation schedule, you'll see red bars there where it got the mass wrong. Okay. So this is something just to be aware of. It turns out I thought naively that Compound Discover pre-filtered the precursor mass before it did the MSMS library search, but actually that's not the case. It's doing the library search and just allows any precursor, which at first I thought, oh, that's not such a great idea, but now I'm glad that the program works that way because it allows us to go back and evaluate compounds that might have unexpected adducts. Okay. So what's the data look like? It looks like this. So this is the heat map of only the 634 library hits across 190 samples. Okay, so it's incredibly dense data. So we're still mining this, and we've been mining this for months. You can see some blocks here. These samples here, there's no way you're ever going to read these, but this is actually the NIST standard reference material dust right here. This cluster of compounds here turns out to be the quaternary ammonium salts, lots of quaternary ammonium salts. These are used as fabric softeners, antimicrobials, etc. This cluster here is made up primarily of non-ionic surfactants, things like octylphenol ethoxylates and nonylphenol ethoxylates, which are actually known to degrade to endocrine disrupting compounds. So both of these are biocides and these are endocrine disruptors. So these are both of concern with respect to exposure. We can prioritize as an example. I guess we're going to talk about some statistical tools. We like these volcano plots, the differential analysis. In this case, we had 7 chemicals that were significantly and twofold enriched in dust from children's homes with asthma. So we were looking at basically with and without asthma. So there's these 7 compounds. There's nicotine metabolite, a cocaine metabolite, and some fragrance chemicals. Now, am I saying that these are causing asthma? No, but these compounds are associated significantly with children's homes which have asthma. And so what's interesting is if we look at cocaine and the cocaine metabolite, these are actually prominent in our list. And what's interesting is that you can, if you plot this versus education level, so this is, sorry, you can't see this, but this is, so this is college graduates, right? There's not a lot of cocaine in their dust. High school grad, GED, only high school is a little more cocaine there. Less than high school, okay, they also have a fair bit of cocaine. And if you have only had some college, I guess you still have some cocaine. So it's interesting what you can mine in this data, and you can learn quite a bit about what people do from their house dust. We found things like fungicides that are used for athlete's foot, so we know who has athlete's foot and who doesn't. We know who uses cocaine and who doesn't in their homes. Okay, so what do we do with the other 10,000 compounds in our data set that had no library matches, right? So we still have 10,000 compounds even after dereplicating. So what do we do with those? Well, so then we're looking at this sort of level 3, and this is kind of the Wild West, right? And this is what we were talking about with MZ Logic and the other tools. So we take a little bit of a different approach to this. What we're doing is we're taking our MS1, MS2. We use the Sirius 4.0 tool that I showed before to get formula assignment. In that case, we check that formula against what Compound Discoverer gives us to see whether they're the same. And then we start to postulate structural formulas. And so I mentioned yesterday, we're using actually PubChem that's annotated with sub-databases like DSS-Tox, HMDB, Drug Bank, pesticides, etc. And then we pass our data here to rank these postulated structures using a variety of in silico MSMS tools. And so what we've done is develop this tool we call Painter, Probability Assessment for Identification in Non-Targeted Environmental Research. So we decided to get cute and come up with an acronym for this. But what it really does is it's an engine for running METFRAG, CSI, Finger ID, Magma, and CFM ID. All of these are in silico fragmentation algorithms that are open source. CFM ID and CSI Finger ID are machine learning based. METFRAG is combinatorial and Magma is rules based. So this is, you can think of these as similar to Mass Frontier, but you can run them as an engine. We take all of that together with metadata, for example, numbers of patents and numbers of references, and we put it together in a generalized linear model, and then this allows us to rank our compounds. And so what this shows is actually the rank of the correct structure using, if we get the percent, basically cumulative fraction, so first rank is the correct, where the correct answer is at rank 1. We had almost 75% of our test compounds after the training of this model gave us a correct first rank of 1. And we compared that to all of the individual fragmenters alone. And so our approach does better than simply the sum of the parts. So we're able to do pretty well with that. We tested this with a variety of different library spectra and trained it with basically about 1,500 high-quality MSMS spectra, and then we're able to evaluate our false positive and our true positive rate. When we include our metadata, for example, the compound or the references, patents, and sources counts, we're getting about a 97% true positive rate and a 2% false positive rate, which sounds great, except for when you look at how many true positives we had. There are 1,400 compounds, 1,400 spectra, there are exactly 1,400 correct answers. But there are not 1,400 incorrect answers. There are 3.3 million incorrect answers, right? Because that's the number of formula or structure candidates that we're assessing from PubChem. So 2% false positive rate is still a fairly large number of false positives out of 3.3 million. Okay? So we're trying to figure out a better way to basically reduce this. And so we need better training data in order to pull these graphs as far apart as possible with this. So just real quick, I just want to show how we've actually used this. Which compounds are we missing with library analysis? So this was an interesting paper that was published a couple years ago where actually they found all of these formulas. This group found a bunch of brominated compounds in house dust, but they couldn't actually assign structures for these. They had no idea what these were because they basically had no library spectra. They analyzed these using negative ion APCI. So it just so happened that we had almost identical samples that we'd analyzed in our lab 3 years ago. So we went in and retrospectively pulled out the data. These were run on actually a fusion system at San Jose. We took some samples and did some tests before we ever bought our Lumos. So what are the structures of these compounds? Well, so we went through our data, and it turns out that for this top rank one, they actually got the formula wrong. So we figured out that what's happening is that these are brominated azobenzene dyes. I'll give away the answer. These are azobenzene-based dispersed dyes. Turns out that in APCI negative, these give radical anions. So they basically undergo electron capture. So these guys thought that they were looking at M-H ions. They're actually looking at M+ electron ions. So that's why, of course, they got their formulas wrong. We figured that out when we did APCI positive. We saw 1 Dalton difference between the two precursors. So we did MSMS on this, and we were able to figure out what this was. This is, I think, dispersed blue 373. We actually were able to get a standard for this and run it by LCMS and confirm it. So that's a level 1 ID. And then we went through and annotated the rest of these. Turns out they got more than just one of the formula wrong. They got a number of them wrong because of this unexpected behavior in the APPI. And so these are a variety of different azobenzene-based dispersed dyes that are used to dye polyester and nylon. And so they've been very, very poorly studied in the environment. There's almost no data about their occurrence in the indoor environment. There's a little bit of work in textile streams downstream of textile plants or waste streams. So why do we worry about these? Turns out that what has been done is there's been studies showing that they're mutagenic in extracts of rivers, and there's this increasing report of contact dermatitis related to textile dyes, unknown textile dyes in airline personnel. And so this is a number of reports of this where they have polyester uniforms, and they get contact dermatitis from areas that are in contact with certain colors of the uniforms. It turns out it's usually blue and black. And those blue and black dyes are actually these dispersed blue 373, 93, and 61 are combined to make the most common black dye in commerce. So we actually wanted to know what other dyes are present in house dust. So we actually went through SciFinder. We pulled out all compounds that had a para-amino azobenzene core that were chlorinated and brominated or non-halogenated. This turns out to be close to 5,000 compounds. We used these to make an inclusion list, isotope pattern matching set in CD. And then we're able to, in this case, get a few dyes that we purified from technical mixtures. Here's where we use our, this is a graphic showing where we use our painter algorithm. So basically here's our MS1, we do MSMS, we annotate all of our fragments here with formulas using Sirius. We take our best fit formula candidate, we pull out all of the compounds, all the structures that have that formula from PubChem, and then we rank them using fragmenter. So here's all the fragmenter scores. Blue or green is high score, red is low score, and then we evaluate the number of references, patents, and data sources. And you can see that we have this one sort of green bar here, and that turns out to be this compound here, which is a chlorinated analog of one of the dyes that I showed earlier. So we're able to actually discover some new dyes using this technique, and then annotate these with structures. So the green ones here, these are all the dyes we found in dust. The green ones we were able to confirm with authentic standards. These are actually percentile scores for all of the fragmenters, Magma, METFrag, CSI Finger ID, and CFMID. You can see that in all cases we had high percentile scores, so this means that Disperse Orange 37 was in the 100th percentile. It's ranked number 1 out of 252 possible candidates by both Magma and METFrag. You can see that CSI Finger ID didn't do so well. So why is that fragmenter underperforming for this particular set of compounds? The reason is because that's a machine learning based tool, and it was never trained on this kind of compound. So these fragmenters do only as well as the data that was used to train them. This is also why it's a good idea to use more than one for these. Okay, so just to conclude, we're using a combination of computational mass spec and cheminformatics to add confidence to these assays, integrating multiple tools and multiple scoring metrics to do non-targeted analysis. And so these azobenzene-based dispersed dyes were just an example of what we can do to identify compounds that we don't anticipate in environmental systems using these tools. So in the future, we're validating this this painter tool rigorously for a lot of different sample types. What we are also doing is combining the library hits that we get with our in silico MSMS scoring. So just because we get a library match in our data doesn't mean we ignore those in our in silico MSMS annotation. We actually do both. And so then what we say is, okay, well, if a compound gives a library match, high confidence library match, and that library match molecule is the same answer that we get in our in silico MSMS of all of our structural candidates, then we give that a boosted level 2 library match status. So that's a Szymanski level 2+. So we're doing computational transformation product prediction. So one thing we also do, we'll take that list of library matches and we'll take all of those and process them through a tool called biotransformer, which you can actually use to specify transformation product rules, transformation rules. So either using environmental or human gut or cytochrome P450 mechanisms. And then we screen, go back and retroactively screen our data set for all those transformation products. So I'll thank my team. People at Thermo that have helped out quite a bit over the years are Ralph and the CD team especially. And then, thank you for your attention.